Great, thank you. Well, it was great being able to follow Joe with the wonderful discussion and background on allergens. I'd like to thank the organizers for the invitation to speak here, and also want to acknowledge my collaborators, John Reichert, who has done enormous work on the extraction of the data, and Bruce Allen, who has been doing the modeling. As noted, this work was funded by ILSI North America. The information presented here, however, is the opinions of the authors and not necessarily the ILSI staff or member companies. I also do want to note that I have done other work on risk assessment related projects for a number of food companies and food company consortia, but none of that was related to allergens. So I'll be talking today, first some background on the data that we used in our analysis approach, results to date, and then our remaining analysis plan. So the overall goal is to be addressing the question about the dose response for an allergic reaction to peanuts in sensitized individuals. And this work was motivated in part at least by a National Academy of Sciences report that noted that thresholds do exist for allergens, and a lot of our work has been modeled after work that Joe Baumert and Steve Taylor and colleagues have done for a variety of allergens. And so our goals are to describe the distribution of individual thresholds for use in the development of a reference dose, to identify the dose associated with several different eliciting doses. So that's the dose that gives a specific defined response level. So an EDO1 would be the dose that gives a response in 1% of the tested population. And our goal is to calculate these different EDX values and then provide that information to regulatory agencies so that they can make informed decisions about acceptable limits. We're accounting for a variety of different sources of variability, and our goal is complete transparency. So when we publish the data, we'll be both providing the underlying data that is used as well as the underlying model code. This is the general design of the studies that provide the data. We were able to get in touch with allergen researchers with the help of the National Institutes of Health. And they put us in touch with researchers who were doing work on desensitization. So they were treating people, and they were looking at this active part of the active arm that they had reached, the patients who had received the desensitization treatment. But the data that we were interested in was from the baseline oral food challenge, and then people who received the placebo treatment arm, and then some of them received an additional oral food challenge. And the way these oral food challenges work is that the individuals are exposed to increasing levels of the peanut protein until there is a challenge-limiting response. So each individual has been exposed up to different exposure levels. So, and all of the people who were tested have been confirmed to be sensitized based on objective measures. They were excluded if there was a prior history of severe anaphylaxis, but we don't know, so that's based on severity of response, not the dose that was tested. So we don't know if the dose that caused that severe reaction was a whole peanut butter sandwich or a very small amount. We don't know whether the most sensitive part of the community was excluded. Some people may have not wanted to undergo this treatment and the challenges because they were concerned about undergoing that oral food challenge. On the other hand, some patients may have volunteered for the desensitization therapy in order to be desensitized because they were sensitive. So this is the summary of the data sets we had from 3 different studies. COFAR stands for the Consortium of Food Allergy Research, and then the Stanford work was done under Dr. Kari Nadeau. The Stanford work had by far the largest number of participants. Both the Stanford and COFAR 6 included a number of young children, and then there was quite a dose range. Note that these are the cumulative doses across the entire oral food challenge. As I noted earlier, all of the subjects had been confirmed by objective means to be sensitized, but not everybody reacted in the oral food challenges, perhaps because they were not tested up to sufficiently high doses. So all of our analyses were based on cumulative dose of either peanut protein or the placebo oat protein challenge. We focused our analysis on the challenge limiting dose, where the study termination was based on the clinician's judgment, but most of those were at least the objective responses. There were a few subjective responses, such as nausea, when they were considered severe enough to be terminating the challenge. In addition, because there were concerns about the potential for severe reactions at the challenge limiting doses, our analysis, we also conducted an analysis based on the dose prior to the challenge limiting dose for challenges that had severe reactions, where severe was based on the definitions of Zhu et al. These severe reactions were either individual reactions of wheezing, or they could be cardiovascular, although we didn't have any of those responses. And also if the individuals had effects on 3 different systems, such as skin, respiratory, and GI. And for both of these types of analyses, we identified the dose prior to the response, which is like a NOEL, and then the dose at the response, which is like a low-L, the lowest observed adverse effect level. We used a survival analysis to do the modeling, so this is different and a little bit more challenging than classical animal testing, where you have each group is exposed to a certain dose level. Here we're modeling each individual's response, because each individual is tested up to different dose levels depending on their reaction. We used Bayesian modeling Bayesian analyses to do the modeling. I'll talk about that a bit more in a couple of slides. And then we used a hierarchical approach to account for different types of variability. So there was variability across the 3 different studies, among the centers within the study. So COFAR includes 5 different geographic centers, and Stanford actually included several sub-studies, some of which were done at Stanford and some at a variety of places. And then there was also variability across the individuals. And in tonight's poster session, I will be talking more about the methods, but what the best predictor in these different levels of hierarchy was accounting for the centers and the individuals. So this shows the association for those individuals that were tested twice, the degree of association between the first NOEL and the second NOEL. So this does not show all of the NOELs for the people who were tested twice, because some people responded at the lowest dose, and we don't have a NOEL for them. And there's a few key take-homes here. The first is that there generally is a correlation between the 2 tests, but there are a number of outliers from a number of different, where the difference between the 2 tests is a couple of orders of magnitude. And then there also is no clear trend for whether the NOEL at the second dose was lower or higher than the NOEL at the first testing. So as I mentioned, we did Bayesian modeling analysis, and the reason we use this more complex mathematical approach is that it provides a way of capturing the human reasoning and biological intuition. It also eases the computational burden of estimating a large number of parameters and the hierarchical relationships. We also used model averaging that considers multiple models simultaneously and is useful for addressing uncertainty at the model level and decreases the impact of the model uncertainty. So rather than having to decide, is this model the best model or that model the best model, it combines the results of all the models, and poorly fitting models get low weight. So this is the results of our analysis using 5 different models that were fit to the data. You can see that the results are generally similar across the models, although there are some differences, and you'll see that more specifically when I show you the numbers. For the observed data, we're showing that in 3 different ways because we have a no-L and a low-L, but the actual threshold is somewhere between the no-L and the low-L, and we don't know exactly where it is between, so we looked at the average between the no-L and low-L. So the blue dots are the no-Ls, these gray dots are the low-Ls, and then the yellow dots are the average between the no-L and the low-L. And this kind of stair step between the doses is what is common for this sort of survival analysis because we only have data at certain specified doses. I also wanted to point out that the x-axis is in log dose. So if we showed it linear, it would look like a much steeper dose response. And also that in this low dose range of, basically we're most interested in the response at 2 milligrams of protein and lower. That's where the 1% response has been from previous work. And that there's good model fit to the data there, so that's good. There's also not a lot of subjects responding, so that increases the uncertainty, and that's also part of why we are interested in having the modeling. So here are the EDX values. So the ED01, 1% response, 5%, and then 10% responses. These are the doses that correspond to those response levels with the confidence intervals. And you can see we looked at several different models. When we included the Weibull model, that actually got all of the weight. And so the ED01 was 0.22 after taking into account all these different levels of the hierarchy. These are some results that are fresh off the press, actually just calculated last week. We found that the Weibull model, as I noted, was the primary driver. We are confident with the EDX values for all these models, but there were some uncertainties about the relative weightings based on the differences in fit. So I'm also, in the interest of transparency, showing you the results without the Weibull model. And you can see that the Bayesian model averaging result for the ED01 is 0.42. So the range of our results right now for the ED01 based on the cumulative dose is 0.22 to 0.42, which is right in the range of previous published results. There are some differences between our methods and the data we used from the previous work. We had 2 primary collections of studies that used a standardized approach at different locations or different trials. In contrast, my understanding of the TNO Nebraska work is that they use data from individual studies, each using their own protocol as well as their own data. There is ongoing unpublished work from the FARC group, which did use Bayesian model averaging. I have not seen any of those results, but my understanding is that they did not include as many levels of the hierarchy. And they also modeled, at least the more recent work, focusing on objective responses. We do have several additional analyses underway. We have looked at the impact of taking into account the responses at the placebo, either ignoring it or two different ways of taking into account. There was no impact of that. As I mentioned, we're also doing an analysis based on the dose prior to the challenge limiting dose for those with severe reactions. And then also working on stratification by sex and age, a comparison of the dose response by severity, and I invite you to come to the poster where we're looking for input on that, where there the question is, if there's a response at the low dose, is there a concern that it would be a severe response? And then finally, we have an additional data set that has been previously identified as high sensitivity, and we're looking at them to determine whether there's a discernible difference in the overall dose response or the severity of response compared to the rest of the population that's been modeled. So the key takeaways are that there's a substantial variability in response on repeat testing. The EDO1 that we have calculated is generally consistent with prior work, and our modeling accounted for model uncertainty and variability across studies, centers, and individuals. So I'll thank you for your attention and take questions in the panel discussion.
All right.